Today's lecture is about the Human Genome Project. The Human Genome Project began they began talking about it in the 1980s and began seriously working on it in the 1990s. The idea was to sequence the entire human genome, which includes the coding regions for all the proteins and RNAs like we've been talking about all semester, and also the non-coding regions, which we've also talked a little bit about, and that would include introns in addition to the exons, but also the intervening sequences that might be control regions for all the genes. The cost for the human genome was approximately $3 billion, at least initially, and you can find out more background information by following this link at the bottom there, genomics.energy.gov. When the Human Genome Project began, there was a fight between the National Institutes of Health and oddly enough, the Department of Transportation about who would get to fund this, and it actually ended up being funded by the Department of Energy rather than the Department of Health. Just an odd twist of history. But then after the project began, there's nobody in the Department of Energy that had the expertise to finish it, so all of the control and the actual execution of it went to the National Institutes of Health. Okay, so what I already briefly introduced this, but this image here shows you what is included in our genome. This would be all of the nucleotide information contained within our 22 pairs of autosomes and our sex chromosomes. So the X chromosomes and the X and the Y chromosome. The material that was used to generate the genome was provided by volunteers, and that information was protected and actually made secret even to the scientists. So it's thought that the genome that we have and that we use as the reference sequence is a compilation of 16 different people. And the volunteers came from all over the world. So it is hoped that the genome faithfully represents the possible genome of humanity that could be from every corner of the earth. Oops, my little popped up here. Sorry, this is a rough slide. That I had a little quiz section here, but they're all going to be answered already. So you can just see the answers. So in individual cells, the human genome is located in the nucleus. I know you guys already know that, but I just wanted to make sure and point out here in this cutaway of a cell, there's the nucleus on the top right around 2 o'clock. And the Human Genome Project dealt initially with genomic DNA from just the nucleus. There is also a genome in the mitochondria, and so since, of course, the mitochondrial genome has also been sequenced, and that information is very useful also. In plants, chloroplasts also have a genome, and those have been sequenced for many different plants. Now we've been dealing with sequences like this. This is a messenger RNA sequence for the cystic fibrosis transmembrane conductance regulator. So the receptor that when they're in your gut, that when there's a mutation causes cystic fibrosis. The human genome provides this kind of information. So just strings of A's, T's, C's, and G's. We've learned already in this class to decode it. So we're using the genetic code to find the genes hidden within the human genome, but also to look for areas outside those genes. So here's some stuff you're already familiar with, taking a DNA sequence or an RNA sequence, decoding it to find the genes or the protein coding region. So another quick quiz question. This one was to ask which organism has the greatest number of genes, from the organism with the greatest to the organism with the least. And the answer for this particular quiz here is that rice has more genes than humans, and we have more genes than yeast. It wasn't always thought to be so. The initial supposition was that the humans would have the largest number of genes. And so way back in the early 2000s, it was thought that humans might have as much as 100,000 different genes based upon the fact that they'd already sequenced the bacteria. So you can see on the top part of this chart, you can come back and look at this later at your leisure. there's around 4,000 genes in E. coli. And if you look at C. elegans, which is a popular model system in molecular biology, it has about 19,000, almost 20,000 genes. The genome was finished for those guys in the late 90s, and so they provided the sort of the basis for guessing how many genes humans had. And so people said, well, we're that much more complicated than worms. We must have a lot more genes than worms. Well, it turns out that we probably don't have that many more genes. So you can see us down on the bottom. And after the draft of the genome was finished in 2003, it was determined that we have about 25,000 different genes. Only about half of those really have been discovered by now. So the goals of the Human Genome Project were to do several things: to sequence the entire genome of humans and also other organisms, model systems such as the mouse or C. elegans or Xenopus and these other animals. It was also determined that the sequencing needed to be done at a very high level of accuracy. Um, In fact, they were shooting for less than one error in every 10,000 bases. And that is absolutely necessary, as I'll show you on the next slide, because how can you determine an error in the sequence from natural variation between different humans? Humans are about 99.9% identical to each other, and so that is one in every thousand bases are different between any two random people. And so how can you tell that from an error? If your error rate is 1%, then you already have a higher error rate than the natural variation rate. So that was a very difficult problem to deal with. And also another thing that came out of the human genome was the development of new resources to deal with biology, chemistry, physics, and combine these fields together, which I think is probably the biggest benefit that's come out of it so far, is that science has become much more collaborative than it ever has been before. So the major challenges, the main one that I just mentioned here was that um, how do you sequence such a large genome? The first predictions were that it would take 20 years or so. And if that were true, we would just be finishing now. But it turned out that as the project began, the technology improved so much that the pace accelerated. And so we finished it in 2003. So it's been about 11 years now. Other things changed as well. As I'll show you, some other things contributed towards speeding this project up, and one of them was a competition between two different genome projects. There wasn't just a single genome project. There was a private one, a company called Celera, and then there was a public one that was funded by the Department of Energy. The other issue I already mentioned how accurate should this be? The DNA sequencing error rate is about 1%. So a single sequencing rate back when this began was about 600 base pairs, and there would be 5 or 6 errors or so per run, and so that's a 1% error rate. And as I mentioned, how do you distinguish this from polymorphisms? So the polymorphism in the human genome is about 1 in 500 base pairs, and how do you tell those two apart? And so the answer was the human genome was sequenced about 16 times, not just once. And so they sequenced it again and again and again and kept comparing it to itself. There also exists a large portion of our genome that can't be cloned, and that makes sequencing much more difficult. So maybe 30% of the genome can't be cloned, and therefore you have to get primary sequence. So that's purifying the DNA from the original source and sequencing it de novo. And that's hard to do, particularly when you don't know the sequence, and so that was a problem that took a while to solve. How did they do it? Well, genomic DNA was taken from the volunteers, was broken into smaller pieces, and as I mentioned, there were two strategies. There was one by the public, that was the Department of Energy. We all paid for that one. They used a hierarchical strategy. It's slower, but they based it upon a genome map that was developed. So they first developed a map by breaking the DNA into pieces using restriction enzymes. They built these big libraries, and then it's sort of like having a big book with a low resolution map that they could place the individual sequence on in the positions where they were supposed to go. So it was putting a jigsaw puzzle together, but by using the picture. if you've ever done a jigsaw puzzle, you can do it without looking at the picture on the box, or you can sort of use the picture on the box to help you build the jigsaw puzzle. So the public strategy used the picture. The shotgun strategy did not use the picture. This was the private company called Solera, and it would be putting a jigsaw puzzle together by using a robot that would randomly try to fit the pieces together. Now that sounds crazy, but actually it's much faster, and it worked. The picture on the bottom shows the announcement of at least the initial sequence at the White House. On the right is Francis Collins, who was the director of the National Institutes of Health for a while, and on the left is, my goodness, I'm blanking on his name, but he was Craig Venter, who was the leader of Solera. He started the company Solera to do this privately. So the hierarchical strategy is shown here in this diagram. You take the genomic DNA, you break it up, and you put it into these bacterial artificial chromosomes libraries, and then you map it. So you have overlapping segments, and then you take each one of those backs and you break it into even tinier pieces using restriction enzymes, and then you sequence each one, and then you can logically put them back together. When you do that, then you work piece by piece by piece, and you keep track of where you are, and you try to slowly put the sequence together. I jumped ahead, and let me jump back to this. I don't show you a graphical picture of the Solera sequence, but the shotgun strategy would be you don't use the hierarchical back library. You take the genomic DNA and you skip all the way down to the shotgun clones. You break it into tiny little pieces and you sequence them. Then you put all that information into the computer and you allow the computer to put the fragments together. People didn't think that would work, and they thought it would generate too many mistakes, but it turns out there is enough variation in the sequence that it worked just fine. So this is a little primer on sequencing. You guys have learned about this before. This is dideoxy sequencing. Now is considered to be old-fashioned sequencing. Most sequencing is probably still done this way, but newer methods of sequencing have been developed. So in order to do dideoxy sequencing, you need a primer with a free 3' end that matches your template DNA. And then you extend your DNA sequence towards or in a normal way using DNA polymerase. In the sequencing reaction, you also have to include the building blocks, which are the deoxynucleotides, A, C, G, and T. In the reaction, you also include dideoxynucleotides at a very small ratio. Dideoxynucleotides are missing the oxygen on the carbon number 3, and so they can't be extended. When they get incorporated, that stops the sequence. And so if you put them in at a low ratio, then the sequence will be stopped every time one of those is added. And if they're there at a low proportion, they'll get randomly added all along the chain. Also, the dideoxynucleotides are labeled with a fluorescent dye, and so you can determine when an A is inserted by the fact that you see a red dye, for example, or you can tell when a C is inserted because you see the blue dye. And then that is run out on a gel so that the pieces of DNA that are generated are separated on the basis of their size. And then that's detected by a reader, a plate reader, or a laser that can illuminate these, and it's read by a sensitive photomultiplier tube. This is what the data looks like shown in those charts. It comes out as light intensity in the different wavelengths, and then it's interpreted by the computer to show you where's a A, where's a G, where's a C, where's a T. And if you look at the sequence, the one on the bottom is actually fairly well resolved. So what that means is you can tell for sure that you have a T or a G or an A or a C. But look at the one on the top, particularly on the left. It's just sort of a jumbled mess, and this is where the mistakes come in. The wavelengths of light are not always 100% completely separated, and the bases don't resolve themselves well, so you can generate error by reading the sequence, and that's why you have to do it multiple times. So the draft sequence was announced in 2000. There's a Craig Venter again and Francis Collins. It was a big deal. I remember all that. It made the news, all the big magazines, which many of them don't exist anymore, but it was a big deal. Approximately 2% of the human genome encodes proteins. This was learned from the Human Genome Project, and that was somewhat of a surprise because proteins do all the work. Other facts. Our genome contains 3.3 billion bases. It's double stranded. We knew that already, but the information content is 3.3 billion bases. The average gene size is 3,000 bases. The largest known gene is dystrophin. It's 2.4 million bases. What I find interesting is that about 42% of known genes have no known function, which is interesting, isn't it? Somebody's got to figure all that out. The human genome sequence is almost exactly the same in all people, 99.9%. So you randomly take two people out of our class and compare yourselves together, and you guys would be virtually identical. But if you 3.1% of 3.3 billion is still in the millions, so there are millions of differences between each of us, but percentage wise it's very small. And then, as I already mentioned, about 2% of the genome encodes for the synthesis of proteins. Now, this pie chart here is the entirety of the human genome, and this shows you the categories of the genes that have been sequenced and discovered, the categories that they fall into. So one of the largest categories of gene function are binding to DNA, so nucleic acid binding. That's a big proportion. Also signal transduction, and then enzymes. Anyway, this is an interesting chart. You can come back and look at it. There on the bottom right, the molecular function is unknown for almost 42%. There's your job security and stuff for you to figure out for the rest of your career. Chromosome 1 has the most number of genes.